welcome back to Botany Fun. That is the first year's botany class. So today we are seeing the LAQ. That is in the first LAQ that is coming from the 5th chapter we have finished in the previous classes. That is the root modifications we have finished, stem modifications we have finished, and along with that racemose inflorescence we have finished. That means out of these three, one is damn sure in the IPE paper for us. Now we will go with the second LAQ, and this LAQ, those who are going for the improvement exam for the first years, they can have it. They can know about this. At least if the supply students also, if you have prepared with the 3 questions from this chapter, that is the sexual reproduction in the flowering plant, that is the chapter 7, according to the Telugu Academy book reader, I am saying. So in that, at least 3 questions, we can guess that 90% we will get one of that question. The other 2 also we have to prepare, and that is only for the people who are going for the improvement exam. For the supply students, this first 3 LAQs, what we have discussed till now, it is the root modifications, stem modifications, and the racemose inflorescence. These are the most important one. From that, one LAQ is damn sure. Now we will go with the easiest and the most important LAQs from the chapter 7, that is sexual reproduction in flowering plants. So in that, the very first LAQ is structure of embryo sac. Structure of embryo sac. Here, they will ask the question, describe about the embryo sac structure, otherwise, whatever may be. So, in that, the first and the answering technique, that is the first one, what we have to write is the structure of embryo sac. Okay. So, now, after this, draw the diagram. So, see how I am drawing. Embryo sac is in the oval shaped, egg shaped structure. So, first draw the oval shaped structure. Then it is having the 2 layers. 2 membranes are present. So, we have to draw the 2 membranes. And in that, the topmost inner membrane, we can close it. And the down portion, that has to be kept like this open, so that it can be, we can go with the drawing the other filiform apparatus very easily. Okay, now we have to draw the ends for this embryo sac. First of all, if you know the ends, then the cell organelle, that is the organelles in this, it will be very clear. The downward portion where we have left an opening for the membrane, that is the micropylar end. Micropylar end. And the opposite one, that is the topmost portion of the embryo sac, that end is the chalazal end. If we know this, then we can go very easily in labeling as well as in describing this embryo sac structure itself. So here we have to draw the cells that are present in the embryo sac at the chalazal end and at the micropylar end. like this, if we draw the diagram, then clearly we can give the labeling. And if we remember the labeling and the diagram, we can explain the structure of the embryo sac very easily. It's very easy. So the diagram we should know first so that we can give the labeling part and the other respective parts. Okay, now start giving the labeling. So at the chalazal end, we will see 3 cells which are present adjacent to one another. Those are the antipodals. Antipodals. And the central one that is just below the antipodals that is present in the blue color, what I have drawn. Okay, that is the polar nuclei. And below that, this is the egg cell. And below the egg cell, the 2 cells which are present in the yellow color, those are the synergates. Synergates. And in the synergates, I have drawn the finger-like appendages. Those finger-like appendages are called as filiform apparatus. Filiform apparatus. Okay. Now we have to give the complete description of this embryo sac. So once we have seen the embryo sac like this, now we have to go with the descriptive part. So the embryo sac. Basically, we can divide, while writing the theory, we can divide the embryo sac parts into 3 major parts. That is the egg apparatus, the first one is the egg apparatus. Second one is the antipodals. And central cell. Like this, we can divide the 3 parts in the embryo sac. So the first one is the egg apparatus. What is meant by egg apparatus and what we have to write over here? Egg apparatus is the one what we have seen. Egg apparatus means egg cell and synergates together is considered as the egg apparatus. So that is present at the micropylar end. So remember, the micropylar end is having the 3 cells in them. That is the 3 nuclei. Okay, that is the one is the egg cell and the 2 synergates. Now, what we have to write in the descriptive part, the egg cell is the egg apparatus is consisting of 2 synergates and 1 egg cell. And these are placed at the micropylar end of the embryo sac. The embryo sac is covered first. So now we have to say about the embryo sac also here. Embryo sac is covered with the 2 membranes, outer and the inner membrane. Okay, and it is having 2 poles, micropylar end and the chalazal end. Now we have to come to the description of this egg apparatus. The 2 synergates are placed at the micropylar end, covering the egg cell behind them. Like this, the egg cell will be present. This is the egg cell and these are the synergates. So the two synergids will protect the egg cell that is present at the micropylar end. And these two synergids are having the finger-like inward growths from the inner membrane of the embryo sac. So that is called as these finger-like appendages. Otherwise, the growths are called as the filiform apparatus. Okay. And what is the function of these synergids and the filiform apparatus? That's the one what we have to write at the end of this. Okay. Actually, at the end of this answer, we have to write the functions of the synergids and the entire egg apparatus and the filiform apparatus also. Okay. So here I'm saying that also because we have started discussing about this now. So I'm saying the functions of the synergids. So synergids will give the protection for the egg cell from the unwanted entry of the pollen tube into the embryo sac. That is the first function. And this will help out in the proper fertilization process. And the filiform apparatus that is present inside the synergids will absorb the nutrients from the nucellus tissue that is present outside this embryo sac. Okay, because this embryo sac is present in the ovule. So in the ovule, again, nucellus tissue is present. So that nucellus tissue is giving the food, that is the nutrients for the egg cell. Those are absorbed by the filiform apparatus. So the functions of the filiform apparatus is to absorb nutrients from the nucellus tissue and supply to the egg cell. That is the first one function for the filiform apparatus. One more function for the filiform apparatus is they will make the easy entry of a pollen tube into the embryo sac. So here they will, whenever the pollen tube has to enter into the embryo sac for the fertilization, then it has to passes through this filiform apparatus. So it will release a such type of enzymes and it ruptures the tip of the pollen tube so that they can release the male gametes in the embryo sac. So it is not guiding. It makes the free entry of a pollen tube into the embryo sac. If they have written in any material, it's a guiding. It's not the guiding. It's the makes the free entry of a pollen tube into the embryo sac. And not into the embryo sac, not into the synergids again. Because at that time, one of the synergid has to disintegrate during the fertilization process. So it cannot enter into the synergid directly. It enters into the embryo sac and releases the male gametes. So the words should be very clear and the sentence should be very clear over here. The filiform apparatus makes the free entry of the pollen tube into the embryo sac for fertilization process. These are the functions of the synergids and the filiform apparatus together. Now comes the second part in the embryo sac. That is the antipodals. What are the antipodals? Antipodals are the three cells that are present at the chalazal end. So here also we have to say the same thing. Antipodals are present at the chalazal end. They are three in number, arranged adjacent to one another. Okay, adjacent to one another in a straight line, in a horizontal line. All the three cells are placed like this. So the three cells are present attached to one another, adjacent to one another. And these the main function of the antipodals is they are meant to give the space and the nutrients for the further development of egg cell during the fertilization process. This is the one what we have to say for the antipodals. Okay, and all the cells are the haploid cells only. So no need for us to worry that whether they are the haploid or the diploid cells. All are haploid. So whenever we are saying for the synergids also, we can say the two synergids are haploid. Egg cell is haploid. Antipodals are haploid. All three are haploid. Okay. So this is, and what is the last sentence what we have to write for the antipodals? At the time of the fertilization, before the entry of a pollen tube into the embryo sac, the three antipodals will disintegrate and gives the space and energy for the further development of egg cell. This is the last sentence what we have to write in the antipodals part. After that, now comes the last part in the embryo sac structure, that is a central cell. What is meant by central cell? Central cell is the one which is having the polar nuclei in it. The central cell is the biggest cell that is present in the embryo sac, in the center of the embryo sac, and it is having the two polar nuclei in it. When the embryo sac is very young, that is immature embryo sac, it is having two polar nuclei in it. Two polar nuclei in it, and these two polar nuclei are covered with the thin membrane-like structure externally, and that is making them to be placed in the center of the embryo sac. Okay, and after some time, after attaining maturity, the two polar nuclei, the cells which are present will fuse together. Okay, they will fuse together to form only one cell. That is the cytoplasmic fusion occurs over here, but the nuclei are free only. So in one single cell, two nuclei we can see. That is the central cell. Otherwise, the one what is present as the what we can say as a polar nuclei. I said not the polar cell, it's a nuclei. These are the two nuclei. So that is the central cell. Now, after finishing of this part, we have to conclude this with the functions of the synergids. If they ask in the question or not, it does not matter, but we have to write the functions of synergids separately. Okay, so what we have to write, I have already said. Once we are discussing about the egg apparatus, at that time only I said. But now again we have to write now. So what we have to write? Synergids are the two cells which are present, are protecting the egg cell in the front part. Okay, like this. In the front part of the egg cell, two synergids will protect them with the to avoid the unwanted fertilization process. Okay, and the filiform apparatus which is present in the synergids will make the free entry of a pollen tube into the embryo sac. And one more function for the synergids, they will give the nutrients and the space for the further development of the egg cell for the fertilization process. So this much we have to write and wind up this answer. So it is the easiest LQ what we can say in the botany subject itself. So prepare well with the diagram so that we can easily go with the explanation part. Okay, so now we will go with the next question, next LQ. That is the wall layers of microsporangium. So what is meant by microsporangium and how to draw the diagram and how to describe the parts. Here also, it's the easiest technique what we have to follow. Once we know the diagram and the labeling, explanation is very, very easy. No need of mug up the answer and the spellings, nothing. Only the diagram and the labeling. If you explain according to that also, we can easily get full score. That is 8 out of 8 in this answer. Wall layers of microsporangium. So this microsporangium is present in the anther lobe of the anther in the flower itself. So the flower is having the anther filament and the anther dithecous anther. Okay. And if the dithecous anther, this is in the front view. Okay. If we are saying this is having the 4 lobes, how can we see the 4 lobes? If the top view of the anther is having the 4 lobes. So this anther is divided vertically like this and perpendicular to that one division, one more line. So that complete anther is divided into 4 lobes over here. Okay. So in each and every lobe, one microsporangium will be present where the pollen grains are going to be produced or formed. So here we have to use the word as the produced, not formed. Formed only for our thinking. Okay. But once we are writing, it should not be used as the word should not be used over there when we are writing the descriptive part of this LAQ. Okay. Here words are proper. If we are not using the proper words, we won't get the full marks. That's the subject wise only I am saying. Okay. So here the microsporangium wall layers. Each and every microsporangium is covered with the 4 wall layers. 4 layers of the wall layers are present. So first of all, we have to draw the diagram of the 4 lobes anther. Okay. The TS of the anther. This is the TS of the anther. Each and every lobe is having 1 microsporangium in it. Like this, we have to draw the 4 microsporangia. And then in the center, it is having the connective tissue. That is the one. So here, the cells, whenever we are drawing the cells, it should not have the intercellular spaces. Remember, if the intercellular spaces are there, anywhere they can cut the marks for the theory as well as for the entire answer. So here, intercellular spaces should not occur. Then inside this, each and every microsporangium, the microspores are going to be produced from the microspore mother cells. And here, when we are drawing the cells, each and every cell should have a nucleus in it. See, I am putting a dot in the cell. This is the life what we are saying, because they will undergo the mitotic, otherwise the meiotic. If these are the spore, microspore mother cells, then they will undergo the meiotic division. So the nucleus representation is mandatory. Then only we can say those will undergo the division to produce number of cells again. And in 1 microsporangium, we have to show them with the microspores formation. And in another microsporangium, what we will do? We will say the pollen grains are released like this. Okay, this is the just actual look how it is. But here, what was the question for us? They asked the wall layers. Here we have to describe only about the wall layers, but this diagram is also mandatory over there when we are writing, describing only a part of the answer. In this, we are describing about only this part. So when we are describing this part, this will attain some maturity. After that, it dehisces to release the pollen grains. So that part also we have to show in the major diagram of the anther. After that, once we have shown it like this, then this part only we have to show as an enlarged part. So in the enlarged part of the microsporangium, okay, we can draw the curve like this very easily. So I'm showing like this. Otherwise, we can draw like this also. Any way, whatever you feel free, we can draw. Okay. Now the microsporangium, the wall layers are 4 in number. That means the microsporangium is having 4 different wall layers. The first one is the epidermis. Epidermis is the outermost layer for the microsporangium, that is for the anther, which is protecting the anther from the outside environmental conditions. And this epidermis is made up of simple parenchymatous I will write the words over here, what I am using now. The epidermal cells are simple parenchymatous rectangular shaped and placed without intercellular spaces. Placed without intercellular spaces. These are the words what we have to write in the sentence for the epidermis. Remember the order, same order we have to use for the another wall layer also. But there the size matters. But here we are not describing about the size, but the same words, same pattern, same thing. We have to use for the 4 anther wall, 4 layers of the wall. So the small layers, the outer epidermis is simple parenchymatous rectangular shaped cells placed without intercellular spaces. Okay, this is the epidermis. When we are saying the parenchymatous, no need for us to say those are the living cells. Parenchymatous mandatory is the living cells. Okay, and all are rectangular. So here the shape and the size and how they are arranged, what we said, that is made up, that is present without intercellular spaces. So whenever we are drawing the intercellular spaces, gaps should not occur between the cells. Like that only we have to draw, we have to mention in the epidermis. The main function of the epidermis is to protect the anther externally from the outside environmental conditions. And the one more function, the epidermis, which is present at the 2 lobes of the microsporangium. Here the cells are very thin walled epidermal cells are present. And these epidermal cells are meant to split, they will help out to split or dehisce the anther to release the microspores into the outside dry atmosphere. So this much we have to remember and we have to write. If you don't know any other thing, dehiscence word, otherwise say the word as the split. The anther wall splits at that point where the epidermals are thin walled and makes the pollen grains, otherwise the microspores, to release into the outside atmosphere. This much we have to write for the epidermis. Now that session was closed. Now the second side heading in the wall layers, that is the second wall layer. That is the endothelial cells. And these endothelial cells, when we are drawing, these should be somewhat bigger than the epidermal cells, and they are placed again without intercellular spaces. That means the cells should not have any gap in between them. and they are hexagonal in nature. These are the shape is mandatory and how you are drawing is mandatory. And with this, we will get the complete answer. It's very, very easy for us. Okay, so this is the endothelium, second wall layer. Now the second side heading in this one is the endothelial cells. So these endothelial cells will be having this pink colored growths are present. So these are not the growths actually, these are the fibrous like wall thickenings. Fibrous wall thickenings. Okay, and this is the endothelium. Endothelium. Okay, so the second wall layer in the microsporangium is the endothelium. This what to write in this descriptive part. Endothelium is single layered second wall layer that is present below the epidermis, and the cells are somewhat bigger than the epidermis and having the fibrous wall thickenings in them. Those are present in the pink color what I have drawn. Fibrous wall thickenings, which gives the extra support for the endothelial cells to protect the microspores that are present in the microsporangium. Okay, and these are hygroscopic in nature. These endothelial cells are hygroscopic in nature. Hygroscopic. What is meant by this word? Hygroscopic means they are, when the moisture is available in the outside atmosphere, they will absorb and they will swell up. If the moisture is not there, they will lose the water during the transpiration process and they will become dry and shrinks. That is the based on the outside atmosphere, the movement of the endothelial cells will depend. That means expanding, otherwise shrinking the size. So that is called as the hygroscopic nature. So the endothelial cells are hygroscopic in nature. Okay, they will expand, they will bulges, and they can shrink based on the moisture that is present in the outside atmosphere. So with this helps out in the release of the pollen grains into the dry atmosphere. If the microsporangium is like this, whenever it is like this, when it will release the pollen grains? Because somewhere else it needs to split, break up. So that splitting will be done, otherwise helps out with the endothelial cells. Whenever they are shrinking, they will make this epidermal cells to come along with them. So where these two lobes are joined, this is called as the stomium. So at this stomial cells, that means the epidermal cells which are present, very thin walled, long elongated cells at the joinings of the lobes, that is called as the stomial cells. So whenever these endothelial cells will expand and contract, they will make the stomial cells to split at one fine movement so that they will release the pollen grains into the dry atmosphere. Okay, so that is the hygroscopic nature. For that, they will have the fibrous wall thickenings, which will give the support for the endothelial cells. Okay, and next comes with the third wall layer. Third wall layer is the middle layers. These middle layers are many in number, minimum to maximum 7. And what we have to say about this middle layers? Middle layers. Middle layers are present in between the tapetum and the below the endothelial cells, and these middle layers are present in many rows. That is minimum to maximum 7. That means 2 to 7 rows of middle layers are present, and all these are the simple parenchymatous rectangular shaped cells placed without intercellular spaces. Same thing what we have written for the epidermis. Simple parenchymatous rectangular shaped cells placed without intercellular spaces. Same words, that's what I said. Same words we have to use for many times over here. Okay, so the middle layers are present. So what is the use of the middle layers? Middle layers are meant to give the specific shape and the nutrients for the further growth of the microspores in the microsporangium. Okay, and once the microsporangium starts its growth, then these middle layers will disintegrate and becomes a powder-like structure over there. That is the main function of the middle layers. Now comes the last wall layer, that is the tapetum. Last wall layer of the microsporangium is tapetum. And see here, I have not drawn any nucleus in any cells or any wall layer except this. So this is called as the tapetum. Last wall layer for the microsporangium is called as the tapetum. This tapetum is having the 2 nuclei in it, binucleated cells. It's not the multinucleated. In some materials, they have given the tapetal cells as the multinucleated. It's not the multi. Multi means more than 2. But here, number specification is there for the tapetal cells. Remember, only 2 nuclei are present in the tapetal cells. So the tapetal cells are simple parenchymatous rectangular shaped cells placed without intercellular spaces. Then the next sentence is all the tapetal cells are binucleated so that they can store maximum amount of food material in them. That's the main nature of the nuclei. Okay. So these cells will protect the microspores externally, and they will give the nutrients to the microspores for their proper development. And inside this are the microspores. Only till here we have to write. So only 4 side headings, and for each and every side heading, nearly 3 sentences are there. And if you know the diagram, explanation is very easy. Explanation is very easy, but we should know the easy method to draw and to remember the wall layers. For any plant part, external covering is always done with the epidermis, what we know. So here also same thing. So how many wall layers are there? So the first one is the epidermis. Second one is the endothecium. Third one is the middle layers. And fourth one is tapetum. So first one, epidermis. Epidermis is the outermost covering for the microsporangium, which will be protecting the anther from the outside atmospheric microorganisms as well as with the changes from the outside atmosphere. And this is single layered epidermis made up of simple parenchymatous rectangular shaped cells placed without intercellular spaces. Now comes the second wall layer, that is the endothecium. In the what we have to write, endothecium is the second wall layer, which is the single wall layer. It is made up of the single layer. Okay, and the endothelial cells are simple parenchymatous hexagonal shaped placed without intercellular spaces. And in these cells, fibrous wall thickenings are there, which gives extra support for the microspores that are present in the microsporangium. And all the endothelial cells are hygroscopic in nature. And when the dry atmosphere is there, in the outside atmosphere, after attaining maturity, the endothelial cells will shrink and makes the stomial cells to split and release the microspores into the outside dry atmosphere. Now, the third wall layer, that is the middle layers. Here we have to say it as the layers. It's not the layer. Okay, middle layers, plural. Here, grammar also considered. If you are writing middle layer and you are saying them as the 2 to 7 in number, then we won't accept. We can cut the marks to half to 1 mark only for the S letter over there. That much important is the S letter over here. If you are writing the middle layer, then the complete side heading is wrong. So we won't consider it. We will consider it as only the middle layers. Remember, grammar counts over here. Learn to write the grammar properly. That means plural, singular need to be mentioned very clearly. Otherwise, it will be a big problem for you when you are going for the second year long answers in botany, where there we will check out the grammar. Everything we will see. Okay. Third wall layer is the middle layers. Middle layers are present below the endothecium, are having 2 to 7 layers, and all these are the simple parenchymatous rectangular shaped cells placed without intercellular spaces. And these, after attaining maturity, once the microsporangium develops, expands, then these middle layers were crushed between the endothecium and the tapetal layer and becomes a powder-like structure, which helps out in giving the layer-like protection for the microspores which are going to be released from the microsporangium. Okay, and the main function is to give the nutrients and the space for the further development of the microsporangium. And the last wall layer is the tapetum. So what is the tapetum? Tapetum is the last wall layer that is present in a single row of cells, and this is made up of simple parenchymatous rectangular shaped cells placed without intercellular spaces. And all the tapetal cells are binucleated, and the main function is to protect the microspores and as well as to give the nutrients for the further development of the microspores in the microsporangium. Till here we have to write. And after attaining maturity for the microspores, the tapetal cells will split and releases the microspores in the microsporangium itself. And after that, the stomial cells will split to release the microspores into the outside dry atmosphere, where those are considered as the pollen grains. Okay, till here we have to write and we have to draw the diagram. Diagram is very, very important in this, what we have to learn properly. And the next one is the process of fertilization. So this question they can ask in different ways. That means they can ask the question that describe about the process of fertilization. Otherwise, describe about the fertilization in the angiospermic plants. Then what to write? And if they are asking the question like this, define fertilization and describe the double fertilization occurs in the angiospermic plants. Anything they can ask in this question. That is the third LAQ from this chapter. So we will discuss about this question in the next class because time won't be sufficient for me to start this and conclude this answer. Okay, because diagrams are there and the steps are there. So we have to prepare that in a proper manner. All the 5 steps will be there in the process of fertilization. So this occurs only in the angiospermic plants because 2 times the fertilization process occurs and 3 times nuclear fusion occurs. This process occurs only in the angiosperms, not in gymnosperms, because the flowering plants, when they are asking for the flowering plants also, we have to say the process of the fertilization in the angiospermic plants only, not in the gymnosperms. Okay, right? So we will discuss about that, and meanwhile, prepare well with these 2 LAQs, what I said in this 7th chapter, that is sexual reproduction in the flowering plants. There is the anther wall TS, that is the wall layers for the microsporangium, that is the 1 LAQ, and then the structure of embryo sac. Because structure of embryo sac and the pollen grains, what we have discussed now, both will come together in the third LAQ, what we are going to start tomorrow. That is the process of fertilization. So that is the most important. If you learn these 3 LAQs from this chapter, expecting that 1 LAQ has to come in the advanced supply exams. Okay. And the 1 LAQ, what we have learned already from the 5th chapter, that is the root modifications, stem modifications, and the racemose inflorescence. So those 3 will learn in that 1 LAQ is mandatory. And the second LAQ, we may get in this, out of these 3, there is the anther wall, that is the process of fertilization and the structure of embryo sac, wall layers of the microsporangium. Other 2 are also there. I am not expecting that will come in the supply exams. May come, may not come, but if we prepare these 3, then if it comes in that, then well and good. If not, you can prepare with the SAQs and the VSAQs, what we will discuss in the class. So with that also, you will get 45 marks. It's not the border pass 21 marks. I'm making you to prepare and go for the 45 marks paper. Okay. So 45 marks means it's nothing but the first class marks for you. So from the failures to the first class, it's a big deal, but it's easy once you follow the tips of what I am saying in this class. So just to follow this and put some time to go through the material and the reader, then you will definitely get 45 marks in botany. First year botany is damn easy, but how we are writing and what we are writing is the most important concept over here. It's not that to mug up the answer and write over there. If you have not drawn the diagram properly, and if you missed one of the labeling also, the theory will be just checked out very neatly, that each and every point will be noticed over there. That means if you didn't keep the full stop, otherwise the apostrophe, otherwise the sentence is not ended in a proper way, that also counts when you have not given the proper labeling for the diagrams. So the diagrams are very, very important. For that sake only I have shown you directly. Okay. So how to draw and how to give the labeling part and how to write the answer by seeing the diagram. So it's a very, very easy task for us to get 45 only with the SAQs and the specific VSAQs and one of the LAQ. in the botany first year syllabus. Okay, so stay tuned to this same timing, same channel, so that you can have the third LAQ tomorrow. And till that, we'll take a break. Right. Thank you.